Or here. Yes. Okay, well, welcome to today's BNP webinar. This is the last webinar of 2021 before winter break. And our speaker today is Tamara Broderick. Tamara is an associate professor at MIT. She has multiple affiliations, including the Statistics and Data Science Center and the MIT Institute for Foundations of Data Science. Her research interests focus on understanding how we can properly quantify uncertainty and robustness in modern and complex data analysis procedures. In her work, Tamara uses Bayesian inference and graphical models with an emphasis on scalable, non-parametric, and unsupervised learning. The title of Tamara's talk today is More for Less, Predicting and Maximizing Genomic Variant Discovery via Bayesian Non-Parametrics. Tamara, thank you for accepting this invitation. And without further ado, please join me in welcoming our speaker.
Great. Thank you so much for the kind introduction and for having me out. I have one request, if people would be up for it. I find it so much easier to give a talk if I can see some faces. And so if a few people wouldn't mind turning on their cameras, thanks so much. I appreciate it. It's just psychologically easier for me. So I really appreciate it. Thanks. Great. Okay. So yeah, thanks again for everybody for coming, especially at the end of the semester. I think it's a busy time. So today I'm going to be talking about genetic variants. And genetic variants can be useful for understanding disease in humans or studying different organisms, but they can be very expensive to find in practice, especially the ones that we want to find. And so we're going to show a new method to find as many as possible under a limited budget. And this basically ends up just being a really fun application of an Indian buffet process. So hopefully you'll enjoy that as well. Okay, so let me start by saying, what do we mean by genetic variant? And the basic idea is going to be, suppose we have two genomes. Those will be a sequence of nucleotides, A's, G's, C's, and T's. And we're going to say that we have a variant whenever those two sequences differ. Now, I'll just mention very briefly, variants, the word variants is in the news quite a lot right now. So we have like the Delta variant and the Omicron variant. That's actually a little bit of a different usage than what I'll be talking about today. So there, you're thinking of an entire sequence and maybe an entire population as being different. So the Omicron variants are different from the Delta variants. Here, we're thinking of a variant as being every difference in two genomic sequences. And so you could have two sequences and actually have quite a few variants just between them. Okay, so why do we care about this type of variant? The type of variant that I'm talking about today, well, it's really useful for, in humans, understanding things like oncology and disease. And more broadly, if we think about all kinds of organisms, it can be useful for understanding evolution and organism diversity. And so people are really interested in finding these variants. Okay, so why is there any challenge to this? Why don't we just take the variants and analyze them? Well, the problem is we have to find them first, and that can be hard. And so there are two ways that we can find more variants. One is that we can get more samples, so we can sequence more individuals' genomes. Or another one is that we can sequence a genome at higher quality. So if we have sort of a low quality sequencing of a genome, we might not find all the variants there. And if we sequence at higher and higher quality, we'll find more variants. Oops. And so basically the situation here is that, well, we can spend our money on sequencing our genomes at higher quality or higher quantity, and we want to find as many variants as possible under this budget. And so that's roughly what we'll be talking about today. Now, the sort of setup that we'll be imagining is that scientists have already done a little science. So they've done some pilot study, they've completed this pilot study, and so they know a little bit about this population that we're thinking about. And now they're planning a follow-up study where they're hoping to find many variants, as many as possible. And we're trying to help them plan that follow-up study. That's the thing that we're imagining as our setup. And in particular, what they'd like to do probably with this follow-up study is have so many variants that they can maybe run some statistical hypothesis tests. So there's this particular type of variant, a rare variant, that can be especially useful. It's hypothesized to explain much of the biology and heritability of disease, but it's also particularly hard to find because it's rare. And so maybe once they have these, they're going to run a hypothesis test to say how those are associated with a particular disease. And they want to make sure that they have enough variants to have enough power in that hypothesis test or to do some other scientific procedure. And so we want to predict how many variants they'll find in a follow-up so they can decide before running their expensive procedure, do they expect it to be worth it? Now, this has actually been done in a variety of work. We'll talk about some of that today. So a lot of people have already done this thing where you predict how many variants you're going to find in a follow-up based on the variants that you have seen in a pilot study. But what that work does not do is does not allow experimental conditions to change. And so in particular, what we're thinking here is sequencing depth. So it could be that somebody else ran the pilot study, in their lab, and so they have different equipment than I do, and so their sequencing depth is different. It could be that I am running my follow-up study now, but I've changed my equipment for whatever reason. It could be that now I have my fixed budget going for it. I only have so much money I can spend, and I've decided that I'm going to spend it on sequencing a really large number of individuals, so I can't sequence them as deeply. And for whatever reason, I might have different experimental conditions. And so what we're going to talk about today is, to the best of our knowledge, the first prediction method for predicting the number of variants we're going to see in a follow-up experiment based on a pilot when the sequencing depth can change between the pilot and the follow-up. Okay, so once we can do this, once we can make this prediction, then we can actually optimize our budget, because if we can predict for any sequencing depth and any sample size how many variants that we can expect to find, well, we can ask over all the sequencing depths and all the sample sizes, when can we find the most variants? And so this has been done before any data is seen. So people have looked at this idea of choosing the optimal trade-off between depth and sample size in that case, but they haven't done it when we've already seen some data. And so this existing method could be used for something like, oh, I'm going to say what's the optimal trade-off, but it's going to apply to every single data set. And we think that, maybe there might be some things that are specific to different data sets. We might like to tailor our methods to particular data sets. And so we want our trade-off to be population specific. And so what we're going to provide here is a way to make this trade-off using information from the pilot study. Okay, so that's what we're going to talk about today. And it turns out that we're going to use the Indian buffet process to do so. And so just as a roadmap for where we're going in the rest of the talk, first I'm going to talk about a little bit more biological background and this problem of predicting the number of new variants in a follow-up study, given that you have this information from a pilot study. And I'll talk a little bit about how the Indian buffet process fits into this. But remember, I'm going to say basically in this problem number one, we're going to do just as well as existing methods. So there are a bunch of existing methods. We're going to reach state of the art. We can trust that this is going to work well. But I think what's really exciting about using Bayesian nonparametrics here is the ability to solve new problems. And so in particular, the new problem that we're going to address is, what if I want to predict the number of new variants in a follow-up study from the pilot study if the sequencing depth can change? And so here we're going to use a tweak to the Indian buffet process to do this. I want to say a variant of the Indian buffet process, but I can't, because the word variant is being used for something else in this talk. And so let's call it a tweak to the Indian buffet process. And then once we have the ability to make this prediction of how many new variants we're going to find, we're going to use it to optimize a budget. And in particular, our proposal would be basically just using directly the predictions from step number 2. Okay. And then finally, we'll see how our method performs in practice with some experiments. And I'll talk very briefly at the end about some, I think, pretty cool extensions, and I think some work that remains to be done in this area. Okay, so let's start by talking about predicting the number of new variants in a follow-up given the pilot. So in order to do that, let's first talk about a little bit more, what is a variant? So very roughly, and I should just say throughout all of this, I am not a biology expert. We are lucky to work with a biologist who really knows these things. And so anything that's right here is due to him, and anything that is wrong here is due to me. Also, there is a lot of nuance in variants. And so really, I am describing a cartoon at best, and there's actually quite a bit more nuance than what I'm describing. So we'll get a little bit more into the nuance later on, but let me just start with the cartoon. Okay, so our reference genome is going to be a string of these nucleotides, again, G's, T's, A's, and C's. And we can think of the reference genome as the thing we're going to compare to. Now, the reference genome doesn't actually have to be the genome of any particular person. It could be an amalgamation or any particular organism. It could be an amalgamation, but it's the thing we compare to. And now what we're going to do for every one of these locations or locuses in the reference genome is we're going to compare it to the same location or locus in a sample. So we now have sequenced somebody's genome or some organism's genome, and we're going to compare it to the reference genome. And in particular, for every locus, again, every position, we're going to ask, are these two the same? If, in fact, they are the same, then we'll put down a 0, or in this case, we'll make a little white box that represents a 0. And if, so in this case, they are the same, so we put down our 0. And if they're different, we'll put down a 1, or in this case, we'll draw a little black box. So here, all of the positions that have a variant where there is a difference, we're going to have our black box, and at all of the positions where there is no difference, we'll have our white box. And so now we can see in this case, there were 3 variants in this sample relative to this reference genome. And so we can do this for our first sample, and then we can do it for our second sample. So here we're imagining that we have another organism, another individual that we have sequenced their genome, and then we get a third sample, and so on and so forth. And so now on the vertical axis here, we can index it by the sample number. Okay, so now we have this collection of variants for these individuals, and we'd like to say, again, what is the number of variants that we learn over time? And so if we're interested in collecting the number of variants, the order of the columns doesn't matter. And so we could equally draw the same thing in the following order of appearance matrix. And so here we still have the sample number on the vertical axis, and now we're just indexing the variants on the horizontal axis. In particular, we're going to take the first 3 variants are going to be the 3 that appear in the very first sample. Then the next variants, the next 4 variants are going to be the ones that appear for the first time in the second sample. So we have our 3 variants that we've already observed in the first sample, and we'll keep observing them in other samples. But now we have 4 that we have observed for the first time in the second sample. And then we have a variant that we observed for the first time in the third sample, and so on and so forth. And so we have this nice order of appearance representation of our assignments of samples to variants. Okay, and we can imagine that we keep doing this. We keep getting more and more and more samples, and then we can draw, the exact same thing that we have in the lower left, but now in the upper right, where we still have sample number on the vertical axis and variant index on the horizontal axis. And now something that's just, I think, much more apparent in this representation is that we can see that there's this edge. And this edge represents the number of distinct variants that we've found so far by sample number. So, we said that we're plotting at the very end, what are the, by order of appearance, the new variants that we've seen for this sample. And so always the leading edge of this is going to be the number of variants we've discovered so far. Okay, so we can just flip this around, because what we're really interested in is the number of distinct variants as a function of the sample size. And so let's just plot exactly that boundary down here at the bottom. And so we can plot the number of distinct variants as a function of the number of samples seen so far. Okay, so we say we want to predict the number of new variants in a follow-up based on a pilot. So let's say capital N is the number of genomes that we have in our pilot. So these are the things that we observed at first. And so we can basically make this little curve just on the data on the pilot. This tells us how the number of distinct variants varies as a function of the number of samples in the pilot. But it's worth noting that when we're going to do our prediction going forward, we're not just going to assume that we have access to this curve, this number of distinct variants as a function of samples. We actually have access to all of the information. So all of the assignments of samples to variants. In fact, we have all the spatial information if we wanted it as well. Okay, but what we're going to predict is the rest of this curve. In particular, suppose we are interested in capital M new samples. We're imagining that we have capital M new samples in our follow-up. So then our total number of samples will be N plus M across both the pilot and the follow-up. And so we're going to want to predict how many total samples are we going to find, and then we could just subtract the number that we already knew about, and we'll have the number of new variants that we want to find. So the total number of variants we want to find, we'll subtract the number of variants we already knew about, and so we'll have the new variants. And so it's worth noting again here that when we're making this prediction, we're purely predicting the number of variants we're going to find. We're not predicting anything else about the follow-up. We're not predicting the assignments of samples to variants. We're not trying to find that kind of information. It's purely the number. So we have the extra information in the pilot, but we're just predicting the number for the follow-up. Okay, so this is the prediction problem. And so now our question is, okay, well, we'd like to do this prediction. What could we possibly do? And I think for this audience, the answer may be clear, especially if you've ever seen the Indian buffet process. This sure looks like an Indian buffet process. Okay, so what are we going to use? What's a natural model to use here? Well, an Indian buffet process is certainly natural. So just in case anybody is not familiar, an Indian buffet process is just exactly a generative model, a Bayesian generative model for these order of appearance matrices. So for the assignments of samples to variants, essentially, usually when we talk about Indian buffet process, we might use different terminology, but it's exactly a distribution, a data generating distribution on these order of appearance matrices, sometimes called left order forms. Okay, now one important point is we're not going to use just any Indian buffet process here. In these biological examples, and certainly in everything that we've encountered, we expect power laws. We expect that the number of distinct variants grows as a power law of the number of samples. And so we're going to use a power law Indian buffet process. And so that's exactly the idea that we're going to pursue, is using the Indian buffet process here. Seemingly nobody has tried it for this particular problem, even though once you plot it like this, it feels like you must. Now, another way to think about the Indian buffet process that's going to come up later in our discussion is as the marginal of two processes. So first, we're going to run a beta process model to generate the frequencies of the variance. And so we could think of these as being, if I had an infinite number of samples, what would be the probability of observing a variant for one particular variant location, one particular locus. So we're interested in generating those variant frequencies. And we're going to imagine, because we're doing Bayesian nonparametrics, that in fact we generate a countable infinity of them, but only some of them are chosen when we make a Broderick likelihood draw top. And so then the marginal of this Broderick process to get, or so this beta process together with this Broderick process will be our power law IBP. And once we do this, we can estimate the number of new variants in the usual way via posterior predicted mean. At the IBP, this turns out to have like a really nice closed form formula that's very convenient to work with. And so that's great. Okay, so why would we think to use this power law IBP here? Well, one, we're going to talk in a moment about how there is a parametric Bayesian model that people have thought about for this type of problem. But a benefit here is that there's no cap on the number of variants. So you might think, oh, but there actually is, there's a finite length to the genome in the end of the day. In fact, there's a finite length that's even less than that to the number of potential variants. And the problem is that because this actually is a very nuanced process, nobody really knows what the appropriate length is. And so that really has to be estimated. And we don't know that in advance. And so this is sort of the perfect case for Bayesian nonparametrics. I mean, it's almost always the case when we run Bayesian nonparametrics that the underlying thing is actually finite, but so large as to be unwieldy. And so instead, what we'd like to do is recognizing that we're just never going to hit any kind of cap that exists, act as though it's infinite, and just see what happens in terms of our sort of growth rate. Okay, so also what's really nice is once we do that, we can just capture a real life power law growth in the number of variants. So when we use this power law beta process, this power law Indian buffet process, we're able to capture that the number of distinct variants grows as a power law of the number of samples. Now, as I said, and I'm going to say this again in a moment, we're going to see this later on in the experiments, this is going to meet the quality of existing methods. So it's going to beat a lot of existing methods, but it's going to be just as good as sort of the state of the art in terms of existing methods for this prediction problem. And I think what's really the strength here is that we can do these other things, that we can do this prediction when the sequencing depth is changing. And so we're going to see all of this in our upcoming slides. Okay, but just focusing on this problem again, on the prediction with the same sequencing depth, this is a well-studied problem. There are lots of ways that people have approached it. So there's a parametric Bayesian method that people have used. There are so-called jackknife, linear programming, good Toolman style methods. Basically, these ideas take the frequencies, the sort of empirical frequencies of the variants and use them to predict how many variants we'll see in the future. And as I said, we're going to see that our IBP approach matches the state of the art. But where it really shines is being able to do new things that we couldn't do before. So we'll talk about that next. Okay, so why do we need a new prediction method? Well, because we want to do more than just predict when the sequencing depth is the same. Okay, so in particular, when the sequencing depth changes, we expect to get a different number of variants. We expect that to actually change this curve, as we've described here. And so let's talk about that for a little bit and what we expect there. Okay, so first of all, let's reiterate the sequencing depth we think may change between the pilot and follow-up for various reasons. Maybe, again, somebody else ran the, on their equipment to get the data on the follow-up in the pilot, and now I'm running the follow-up. Maybe I've changed my equipment between the pilot and the follow-up, or maybe I am interested in saving budget, and so I'd like to use maybe a lower sequencing depth than I might have otherwise used, so I can sequence more samples. Okay, so for all these reasons, we'd like to predict the number of new variants that we're going to find in a follow-up based on the number of variants and all of the variant information that we have in the pilot. And to the best of our knowledge, no method handles this. Okay, so we're going to propose a tweak to the Indian buffet process to handle this change. And I'll talk about that in a moment. first let's understand exactly what we're expecting from changing the sequencing depth. So let's dig into that a little bit more. So first, let's call lambda the average sequencing depth. We can think of this as sort of the quality of the sequencing. And we're going to see in a moment that as we increase lambda, in general, we find more variance. We have a higher quality sequencing. Okay, so how does sequencing work? Well, earlier we talked about it as though like somebody just gives us the exact genetic sequence and then we compare it to the reference genome and we're done. And the reality is that we actually don't observe the exact genetic sequence. We observe a noisy version of that exact genetic sequence. And so in particular, if we look at one locus, where we could have a variant, we don't just observe one observation or one read, we actually get a Poisson lambda number of reads. And so we can think of D as the actual sequencing depth and sort of lambda is controlling that as an average sequencing depth here. So we have this sort of number of observations, D. Some of those might be errors, so those get discarded. And then the ones that aren't errors, we look at those observations and we call a variant. We say that we've seen a variant based on some amalgamation of that information. So an example of this would be, we might say a variant calling rule could be if a sufficient number of reads differ from the reference, then we'll say that we have a variant. And actual variant calling rules are actually just even way more complicated than what I've just described. But that's sort of an idea of what a variant calling rule might look like. Okay, so what actually happens here then, a way to describe it, is there is or is not a true underlying variant in the actual genetic sequence. And then based on the sequencing depth, we have some probability of actually observing it if it is there. And so let's see this in our illustrations from before. Okay, so before we said, again, we have the reference genome, we have the perfect sample that we observed exactly, and there's no observation error. And so then we got for every one of our samples at every locus, whether we have a variant. And then based on that, we could construct this order of appearance matrix. And so now we're going to change this observation a little bit. We're going to say, hey, for every one of those variants up above, this is the true underlying variance, there's actually now this observation noise. So we say, okay, not only is there or is there not a variant, but now we're going to ask, do we actually observe that variance? So we're going to have like this extra Bernoulli draw. Do we actually observe that variant? And so once we make that draw, we're going to lose some of the variants in our actual observations. They're still there in the actual genome, but we didn't observe them. And so how's that going to change our order of appearance matrix? Well, we're going to have fewer variants that we've observed. And so for the same sample number now, we're going to have fewer variants. And so the thing that we're observing here is that lambda, the sequencing depth, really affects the number of variants that you actually observe in practice. In particular, reducing the sequencing depth reduces the probability of seeing a variant, which in turn reduces the expected number of discovered variants. Okay, so on one side of the slide, on the left side, we're sort of talking about, we have this one lambda, this one sequencing depth that we're applying, but of course, in the setup that we've been describing, we actually have two. We have our pilot lambda, our pilot sequencing depth for our pilot data, and then we have this new study, the follow-up study, with a different sequencing depth. But we expect the same sort of thing, that when we adjust lambda follow, that will adjust the number of variants we see now just in the follow-up. So in particular, we can imagine decreasing just lambda follow, not lambda pilot. And so then the number of variants we see in the pilot will stay the same, but the number of variants will decrease in the follow-up. And similarly, we can keep decreasing lambda follow and we'll decrease the number of variants that we can find. Okay, so we really see here now that there's a trade-off. So to find more variants, we can either increase the sequencing depth lambda or increase the number of samples. So in particular, if I have a particular lambda follow, a particular setting of the sequencing depth for the follow-up study, then you can see that as I increase the number of samples, I'm going to monotonically increase the expected number of variants that I'm going to find. Now, at some point, there might be diminishing returns. At some point, maybe I'll find the most that I could for this lambda follow, or I won't find too many more as I put in larger and larger M, but I do get that monotonic increase. Okay, and then same thing for a fixed M. If I have a fixed size of my follow-up study, then as I increase lambda follow, I expect to find more and more variants. Again, there might be diminishing returns at some point, but in general, I monotonically expect to find more variants. Okay, so now we're going to talk about what is our method to handle varying depth. And I'm just going to give sort of a high-level sketch of why this is nice to do within, in particular, the Poisson point process framework of Bayesian nonparametrics. And so, if anything on the slide is unclear, the takeaway message is just that if you can handle beta Bernoulli conjugacy, then you can handle this kind of calculation. It might just take a little sitting down, and we can do it in this particular case. Okay, so we said earlier that our Indian buffet process is secretly a power law beta process likelihood with or prior with a Bernoulli process likelihood on top. So what we're really doing is we're generating a countable infinity of potential variants, like 1, 2, 3, 4, 5. Let's just label them. And then for every one of those variants, with our beta process, we're generating a frequency, a probability with which it occurs, a number between 0 and 1. Let's call it theta 1, theta 2, theta 3, theta 4, theta 5, et cetera. And so we do that with our power law beta process. And the way that we do that is via a Poisson point process. And so it's described by a particular rate measure. The rate measure is here. It has some hyperparameters, sigma, c, and alpha, which is absorbed in something I'm just calling constant here. Now, what's really nice, or one thing that's really nice about Poisson point processes, is that it's really easy to pick out a subset of their points. And we can do that with something called marking. So with a marked Poisson point process, we can imagine that we go to every one of the values in the Poisson point process and give it a little mark with some probability p of theta. So for instance, if I have all my variant frequencies, I can give each one a mark with probability p of theta, and then I can collect only the ones that got marked. So if I collect only the ones that got marked, that's also going to be a Poisson point process. And it's going to have a new rate measure, which is the old rate measure, density times this p of theta. Okay, and then if I want to know how many got marked, the number of marked elements, that's going to be Poisson distributed with its parameter equal to the integral over this, let's call it new prime, this new rate measure, new prime. And we're just going to put that integral into the Poisson. Okay, so why is that useful to us? Well, let's first think back to the case where the depth, the sequencing depth does not change. So in that case, we want to say, let's take all of our variant frequencies and let's mark only the ones that are new variants in the n plus n sample, that correspond to new variants in the n plus n sample. That is to say, they were not sampled, or they were not detected in the first n plus n minus 1 samples, and they were detected in the n plus n sample. Okay, well, that's going to be a Poisson point process. And by the marking theorem that we just wrote up there, it's going to have a rate measure that we can describe. So it's the original rate measure of all the frequencies times a p of theta. Why is this the appropriate p of theta? Well, first of all, theta is the probability that we have a variant in this particular sample, in one particular sample. So 1 minus theta is the probability that we do not have a variant in a particular sample. Now we're looking at the first n samples, the ones in the pilot. So this is the probability that we do not have a variant, this particular variant, in any of these samples. And then we can ask, well, we also don't want a variant in the first m minus 1 samples of the follow-up, but we do want to pick out the variants that occur that finally are seen for the first time on the n plus n sample. And so this is a Poisson point process describing the frequencies of all the variants that are new, that are seen for the first time on the n plus n sample. And now if we want to say how many variants did we see for the first time on the n plus n sample, we just say, well, that's Poisson with the integral of this value. And the integral of this value is fundamentally straightforward to take, because you can see this is just a beta kernel, like a regular beta distribution kernel, and it'll turn out to integrate to something finite. Okay, and in fact, you can just derive the usual Indian buffet process formulas using this kind of technique. What's useful about this is that now we can take this technique and we can apply it to the case where the sequencing depth changes. So the idea here is you want to say, okay, our sequencing depth for the pilot is lambda pilot. Our sequencing depth for the follow-up is lambda follow. And so we want to say how many new variants appear on the n plus n sample in this case. So again, that's going to be a Poisson point process. We're going to start by saying, basically, what's the probability of even having a variant? And now let's talk about the variants that are observed for the first time on the n plus n sample. So that's going to be this p of theta. It's a little bit more complicated, but we'll talk through it. Okay, so first we have the probability that this sample, this particular sample, even has this variant. So that's theta. Now, lambda isn't directly a probability itself, but it controls a probability of observing this variant. So if we have the variant, now there is some probability modulated by lambda that we actually see the variant, that we actually observe the variant. So let's call that phi of lambda. We can actually write it down exactly. It's based on some Poisson stuff. But for the moment, let's just call it phi of lambda. Okay, so together, this is the probability that now we not only have a variant, but we observe it in the pilot. And we were interested in the probability that we do not observe this variant in the pilot for every single one of the pilot samples. So this is a variant that we have not observed at any point in the pilot. So that's this probability. And so now we said, okay, well, we actually want something that's observed for the first time in the n plus mth sample. So here's the probability that this variant is observed in the follow-up on one of the follow-up samples. So first it has to exist in the follow-up sample, that's theta, then it has to be observed, that's phi of lambda follow. Here's the probability that it's not observed in the follow-up, and here's the probability that it is not observed for the first m minus 1 samples of the follow-up. And so finally, we want the variants that were not observed in the pilot and not observed in the first m minus 1 samples of the follow-up, but were finally observed on the nth sample. And so when we put it all together, we now have this Poisson point process that describes the frequencies of all the variants that are new on the m plus m sample. And so when we have that, if we want to say, oh, what are all the variants that are new up to some point in the follow-up, maybe capital M, well, we'll just say, what are the variants that were new on the first sample in the follow-up and the second sample, and how many were on the second sample of the follow-up, and so on. And we can get that number by just integrating this. So again, the number of variants that will be observed for the first time in the m plus m sample will be Poisson with its parameter equal to the integral of this quantity. Okay, this is all just to say that when we work with Poisson point processes, we can sort of do what almost looks like just regular beta Bernoulli conjugacy. Here, we're not quite conjugate, but we can still do a simple one-dimensional integral that's not too hard and make this prediction. Okay, so now we have this method to handle varying depth. And so once we have this ability to predict what's going to happen when we change the sequencing depth, in the follow-up, we can ask, how can we maximize the number of variants under a fixed budget? So recall, there are two options that we're considering for increasing the number of variants. And those two options also increase the cost to us. So one is to increase the sequencing depth. So that can have us discover variants, but it's also going to be more expensive to do. And the other is to increase the number of samples that we sequence in the follow-up, but that can also be, will be more expensive to do. And so for fixed budget B, we're basing a trade-off between lambda and n. Do we want to spend more on quality or more on quantity? We probably expect that there's going to be a sweet spot in the middle, and we're going to see that later. Okay, and so in particular, what we're going to do is we're going to consider every lambda and m. We're going to predict the number of new variants that we find, and then if we can do that, we can say, okay, over all the lambda and m that we might consider, what's the best choice of lambda and n? And then we can use that to optimize the trade-off. And so we'll see that in a moment. And let's just talk about the experiments that we're now about to transition to. Okay, so what are our experimental goals here? So first, we are coming up with this new prediction method. We want to check that it at least is matching the other prediction methods, even in this case where the sequencing depth does not change. If it can do as well as these other methods in this other case, then maybe we can trust it more in the new case where there's only our method. So when the sequencing depth does change, we said we don't have existing methods to compare to, but we can still check that our predictions are accurate, that they're close to the ground truth when we have a ground truth. And then finally, we can check that we can optimize for the use of a budget B. So here we want to check that really we do get differences in the sequencing depth, and then when we do, we can actually use them to optimize our use of the budget B. Okay, so these are the things that we're going to check in our experiments going forward. Okay, so first let's look at prediction under the same sequencing depth. So we're going to check that our new predictions are at least as good as the state of the art when the sequencing depth lambda does not change. So to do this, so if you actually look at our paper, we look at a lot of different data sets. So in particular, we look at, I think, 33 different types of cancers in the Cancer Genome Atlas, a number of different subpopulations in the genome aggregation data set, the NOMAD data set. And so here, just for illustration, I'm going to focus on one, but if you check out the paper, you'll see quite a bit of others. So here we'll look at this African-American subpopulation of NOMAD. And so here we have just a plot of sort of what we have going in with a caveat. So on the horizontal axis, we have the number of samples in hundreds. And so we're going to go up to around 8,000 samples. On the vertical axis, we have the number of variants in millions that we'll be discovering. And so we have a vertical line at n. This is again the number of samples in the pilot. And we can, because we have all the information in the pilot, we can always draw our curve so far, which is the number of variants observed as a function of the number of samples. Now remember, in terms of prediction, we actually have quite a bit more information from the pilot, but this is just the curve that we're drawing for the number of samples so far. Okay, so how are we going to keep drawing and comparing our methods? Well, we're going to use a setup that's similar to Zhou et al. 2016. So this is another method that makes these predictions. And it's basically like cross-validation. So we're going to split our data into 33 bins. There's no special significance to 33. It's just some number of bins. And then for each one of those bins, we're going to treat that bin as the pilot, as sort of a small collection of data that we have already gotten. And then we'll treat the remaining 32 bins as the follow-up. And we want to predict the number of variants we'll find in the follow-up. So we'll just give our algorithms access to the pilot, the single bin, but then we'll have the ground truth because we have the other bins to compare how the predictions go. Now, to summarize the information across essentially what are 33 different prediction problems, we're going to plot the mean of the predictions for each method and a plus or minus 1 standard deviations to show how much they vary. Okay, so let me talk through what's going on in this graph. So first of all, we have our ground truth. So again, we have ground truth just because we have these other 32 bins. For every one of these bins, we actually can observe what are the variants here, or at least the observed variants. And so we can plot that as ground truth. And actually, we could plot variation here, and it just so happens that there's not too much variation. Okay, now we can look at the parametric Bayes estimator of Laza et al. 2019. I think this should be 2009. Sorry for the typo. So we can look at this parametric Bayes estimator, and we can see that it's pretty far from the ground truth. And we're not getting too close to that. And you can see that this is not in particular giving a great approximation here, whereas our Bayesian nonparametric method based on the Indian buffet process seems to be doing a lot better and getting close to the ground truth. So why is that? Why are we seeing this difference? Well, one, we talked about some challenges of parametric Bayes. So one was you have to essentially estimate the true total number of genomes or variants, and it's sort of unclear what that even means. And two, they don't necessarily encode power laws. The parametric Bayesian estimator basically says there's just the same beta distribution describing every one of the frequencies instead of this beta process. And so with the beta process, we can encode a power law distribution, but with the just parametric Bayes, we can't get that power law. And we actually see that the good Toolman estimator struggles in the same ways as parametric Bayes. It's pretty far off from the ground truth. We find that this is also because it struggles with certain power laws. So we investigated this in particular in a bunch of simulation experiments. So if you have a perfectly specified model, then it's no surprise that parametric Bayes does fantastic. And it just seems to be that in real life, we actually observe these power laws where it struggles. Okay, let's look at another competitor. So here we've looked at parametric Bayes. Now we're going to reset this graph and compare to the linear programming method of Zhou et al. So again, we have our ground truth. Now we're going to compare to the linear programming method of Zhou et al. 2016, which is doing better in this case, but it's still somewhat far from the ground truth, and our BNP method is a bit closer. Okay, so what's challenging about linear programming? Well, we find that it does really well when the number of data points in the pilot is quite large. When the number of data points in the pilot is very small, we find that this linear programming method struggles. And we think that that's a case of interest to biologists where you've spent just a little bit of money to scope out, what this population is like, and now you're planning the big experiment down the road. And so it's not always the case that your pilot is already very large. Another issue that or challenge that we face in this particular case is that there's this tuning parameter that the linear programming method has. Basically, you have to specify which variants or which variant frequencies you're going to count as rare and which you're going to count as common. So you just have to specify sort of a cutoff. Is it 1%? Is it 0.1%? Whatever. And this actually really changes the results. So we found that for some of the default settings of that hyperparameter that we actually didn't get very good results in terms of prediction. And so we had to change those settings to get better results. And sometimes we also got numerical errors. Now there's actually also a linear programming method due to Gravel et al. 2014. And we found that really just sort of gave numerical errors. And actually Zhou et al. 2016 also did not compare to Gravel 2014 for the same reason. Okay, so finally, let's look at the jackknife. So this is actually an idea that in some sense dates back to the 70s in Burnham and Overton's 1978 paper, and more recently has been adapted to sort of an updated version by Gravel 2014. And it has an order associated with it. And sort of the general conception is that order 3 or 4 generally does pretty well. And that's certainly what we see here, that order 3 and 4 are extremely close. And I would say even closer to this ground truth than our Bayesian nonparametric method. And when we look over many, many, many data sets, we find that in general, this is true, or more to the point, that these methods are just basically perform the same. And so here we're seeing that jackknife seems to be the state of the art, that it seems to be doing really well, at least the problems that we looked at. And our Bayesian nonparametric method is matching it in terms of performance, but I wouldn't say that it's necessarily beating it. And so, again, we have our ground truth, we have our jackknife and our BNP method. And so the observation that we want to make here is that, okay, we're meeting the state of the art, that was our goal, but now that we have this flexible Bayesian nonparametric framework, we can do these new things that we couldn't do before. In particular, we can adapt to varying sequencing depth, whereas it's not clear how we would do that with the jackknife. Okay, so let's look at that problem next. So now we want to talk about prediction under varying sequencing depth. So now we want to check that our new method for predicting the number of new variants we're going to get when the sequencing depth can change between the pilot and the follow-up, that it gives accurate predictions. And so we're going to look at the same subpopulation of the nomad data just for illustration as we did before. You can find the other examples in our paper. So again, on the horizontal axis, we have the number of samples in hundreds. On the vertical axis, we have the number of variants in millions. And the big difference here relative to the last experiment is that we're going to imagine that there's a different lambda, different sequencing depth in the two cases. So in the pilot, we have lambda pilot, and the follow-up, we have lambda follow-up. Okay, now how are we going to set up our experiment here? We're going to use a similar cross-validation like setup that we did before. We're going to use the bins and all of that. But one thing that's awkward is that we do not have access to actual biology labs that can sequence at different depths. And so we're going to have to simulate the additional structure due to the different sequencing depths. And so what we're going to do here to get a ground truth is we're going to take the original data set, in this case, the nomad data set, and we're going to add some additional thinning. So we're going to use exactly this observation likelihood that we talked about before, some additional thinning to get observations as though they were not fully realized due to the sequencing depth. And so there is an element of simulated data here that we're going to test against. Okay, so once we do that, we can again plot the ground truth. So it's just this thinned data relative to the original data. We can check that, jackknife order 4 was doing fantastic before, and order 3, as well. And those are all pretty far from the ground truth now, because the ground truth is different because the sequencing depth has changed. Whereas our Bayesian nonparametric method, because it's able to adapt to the sequencing depth, is able to more closely mirror the ground truth. And in fact, to change its predictions as lambda changes, which the jackknife and these other methods are not able to do. Now, I'm focusing on the jackknife here because the other methods we found had various issues, even in the case of predicting under the same sequencing depth. So here we're just focusing on something that we expected to have good performance, but doesn't when the sequencing depth changes. Okay, so now what we'd like to do is we'd like to use this information to choose the best sequencing depth, to choose the best setting of our experiments. And of course, we can only expect to do that if we have a method that updates when the sequencing depth updates. So if we took the jackknife, it has the exact same expectation of how many variants we're going to find, no matter what the sequencing depth is. And so there's no way to optimize that. Everything looks equally good. But now that we have a method that can be adapted to the sequencing depth, we can use that to optimize. Okay, so let's look at this optimal use of a budget. So our question number 3, our experiment number 3 that we wanted to look at was checking that we can optimize for the use of a budget B. Okay, so here let's look at the cancer genome atlas data. In particular, we'll look at a particular type of cancer, uterine cancer. Again, just see our paper for all the variants of these experiments on the TCGA and nomad data sets and the other types of populations or other types of cancers. Okay, so here, what we're seeing on the horizontal axis is the sequencing depth and the follow-up. That's the thing that we would like to control and to ideally set in an ideal way to get the most expected number of new variants. On the vertical axis, we have the expected number of new variants as predicted by our method. Now, how exactly are we getting this curve Well, we're going to use a setup that's very similar to Ionita-Laza 2009. So in particular, in Ionita-Laza 2009, they again did this prior analysis. So they didn't see any data. They said, okay, how can we trade off sequencing depth and number of samples just in any future experiment? And so now we'd like to do this in a way that's adaptive to the particular data that we have in front of us. And so what we're going to do is we're going to say, hey, suppose we have this budget B. Well, for any particular sequencing depth lambda, that's that horizontal axis, there is a maximum number M of samples we can take in our follow-up that's still within our budget B. So here I'm saying, hey, suppose we have an axis, we're able to say what is the cost if we're taking M samples and we have this sequencing depth lambda follow and match it to B. In general, M is actually an integer, and so we might be just a little bit below B, but we'll get the largest M we can do for a particular budget B. Okay, so once we have our sequencing depth, then our M is determined by, we want to make M as large as possible for our budget. And so we can ask for that particular M, that particular number of samples, as particular sequencing depth lambda follow, what is the predicted number of new variants from our method? And so that's what we're seeing as this, this line here, this black line. And so let's just talk through a little bit what's going on in this black line. So if we go far to the right, as the sequencing depth increases very large, then what's happening is, yes, we're getting more and more variants from increasing the sequencing depth, but we're getting sort of diminishing returns. At the same time, we have to sacrifice actually using fewer samples because of our limited budget. And so because we're using fewer samples, we're not picking up as many variants. Conversely, if we look at the sequencing depth decreasing, becoming very small, well, we're able to sample a lot more individuals, a lot more organisms, because we're able to use our budget that way because we're not using it on sequencing depth. But on the other hand, the sequencing depth is so small that we're not picking up as many variants as we could from each individual organism. And so there's this sweet spot marked by this blue star in the middle where we make a nice quality quantity trade-off, that we have a sequencing depth that's sufficiently high that we're picking up lots of variants, and we have a number of individuals that we're sequencing that's sufficiently high that we're picking up lots of variants. And in fact, we can find the optimal lambda star and M star on this graph. And then the number of new variants that we expect from that optimal case is given by the horizontal line. Okay, so let me just go ahead and conclude. So we proposed this prediction method that we think is the first of its kind for suggesting, for predicting the number of new variants in a follow-up when the sequencing depth can change between our pilot and our follow-up study. So lots of people have studied the case where the sequencing depth cannot change. And we find that we have state-of-the-art performance in that case, but now we can also do this additional thing where we're able to predict what happens when the sequencing depth does change. Also, now that we have this, we can provide the first method, again, to the best of our knowledge, that uses pilot information, that uses the information, any information we have about our data, to optimize a budget between sequencing depth and follow-up sample size. So we talked a little bit about where you can find this work and some related work. So first of all, if you're interested in everything that I've talked about today, this is in this paper in Biometrica. So again, the first author is my fantastic recently graduated student, Lorenzo Massawero. He's really wonderful, and now he's at Amazon Research. And we've been so lucky to work with fantastic people that I'm sure many of you know, Federico Camerlenghi and Stefano Favero, who are really just wonderful and are co-authors on this paper. Now, something that I've really focused on throughout this talk is being interested in understanding the number of variants that we're going to get from a follow-up experiment. So maybe predicting the number of variants or optimizing the number of variants. But sometimes we actually want to focus on a slightly different thing. We actually want to focus directly on what are we going to do with those variants. So again, something that people might do in rare variant association studies is say something like, oh, I actually want to run a burden test. I want to find the association between the rare variants and some particular disease or whatnot, but I want to run a hypothesis test. And so in that case, I might not be interested directly in predicting the number of variants, but I might be interested in predicting the power of that test. Maybe I only want to get my follow-up study and sequence all my individuals if I can have a sufficiently high-powered test. Or because I'm interested in the power of that test, I might want to trade off my sequencing depth and my number of individuals in the follow-up, my number of samples in the follow-up, in a way that maximizes that power under a fixed budget. And so this is what we've been focusing on in this new paper, Bayesian nonparametric strategies for power maximization in rare variant association studies. This is very early work. It's just appearing in a workshop right now, a workshop that's just about to happen next week. The Learning Meaningful Representations of Life workshop. It's sort of a bio meets data analysis workshop. And down the road, we're hoping to put this into a journal paper, but again, it's still very early work for the moment. And on this, I just really want to call out, Josh Schreiber has been really fantastic. He's really the bio expert. Again, if I said anything right, it's due to him. And if I said anything wrong, it's due to me. And he's really helped us a lot with thinking about our earlier work on this as well. So we're very grateful to him. This work actually, the work that I just talked about also builds on this idea of, well, actually, when we're doing hypothesis tests, we're often comparing two populations. So like in a disease case, there's an affected and unaffected population. And so for that, it's actually really helpful to have hierarchical modeling. So like a hierarchical beta process instead of just a single beta process, which is some work that we've done in another workshop paper in the past, this AABI workshop. And then, of course, I think something that's very natural for this kind of crowd is to think, well, do we have to use an Indian buffet process? It seems like there's a lot more information that we could potentially use here. So in the Indian buffet process, that I talked about today, and actually in every one of the methods that I talked about, the good Toolman, the jackknife, the linear programming, all of these methods treat the different loci as though they are unrelated, different variants as though they are unrelated. And so you're basically throwing away spatial correlation. We're throwing away what's known as linkage disequilibrium in the biological lingo. And there's other information that we're throwing away as well. And so it seems like maybe if we could use that information, we could do even better. And so we've started a little bit of a thought in that direction about how we might do that, but I think there's just probably so much more that can be done. And so we have this early work that's up on the archive here. Okay, again, thanks for showing up at the busy end of the semester. Really appreciate it. Thanks for being here today.
Okay, so let's thank Tamara for this presentation. And well, we now have time for questions. So if there are any questions or comments for Tamara, please turn on your mic and go ahead, or you can also use the chat to ask your questions here in Zoom.
I'll ask a question. Tamara, a fantastic talk. Are issues with sequence alignment at all relevant here? Insertions, deletions, etc.?
Yeah, so I think, to the best of my knowledge of sequence alignment, which I think again we're reaching some of my biological shortcomings, but I think that those really come up in this question of what exactly is going on in variant calling, and so in an actual variant calling rule. So in the way that I described this, and the way that we conceptualize it in the paper, we imagine that there truly are just these Poisson number of reads that you get. And then for those reads, we ask, do you meet some threshold in terms of determining them? And I think that when you actually include all of these additional nuances that arise, for instance, in sequence alignment and other things, then you have to have a much more complicated variant calling rule. And to be able to incorporate that into a method is sort of an interesting question. I mean, one thing that's really nice here is that by using the variant calling rule that we do, we have this really nice way of describing phi, the probability of observing the variant given the sequencing depth. And so that just makes sort of all the math work out really easily. And I think, though, that if you could describe phi given lambda, possibly even in an automated way, then you could still go forward with the sort of Poisson point process marking description that I gave. And so it might be interesting to think about, even if there is all this additional nuance, could you just do something empirical to make this work out? And so I'm not saying for sure this idea is going to work, but this is my thought off the top of my head at the moment of this conversation. Hopefully that helps.
Great, thanks.
Thanks so much. I see that there is a question in the chat. I'll just read it and in the process hopefully understand it. Do you think there are natural ways to extend the jackknife approach for different sequencing depths when the pilot study contains samples of varying known depths? I think this is a super interesting question. So we tried to give it some thought while we were doing this. We were like, oh, could we extend these other methods? And I think it's super straightforward to extend the Bayesian method, the parametric Bayesian method. I mean, you just use exactly the ideas that we were talking about here, but with parametric Bayesian instead of non-parametric Bayesian. And we just didn't bother to do that because it seemed like the parametric Bayesian method just wasn't performing at the same level as the non-parametric Bayesian method. So, might as well just do it with the non-parametric Bayes. And then sort of similarly, we didn't focus as much on Good-Toolman or the linear programming methods because again, it seems like they're not quite meeting the state-of-the-art performance even in the baseline case. But I think you're totally right that the jackknife is meeting that baseline performance. And so maybe by incorporating this kind of information in the jackknife, it could do really well. And it's just not really obvious what to do in the jackknife. So the jackknife, I think, is a little bit of a somewhat perhaps misleading name in this case. So when I think jackknife, I think immediately of like the cross-validation variant jackknife. And here it's more like there's some kind of prediction that's based on the observed frequencies. And it's just, I mean, there's some connection there, but I think it's not just immediately obvious what that connection is. And so this is all just to say that it's not immediately obvious, but it would be an interesting question. And if somebody solves it, I'd be super interested. So let me know. I think I see a question.
Sorry, do we have another question from Patrick? Please unmute and go ahead.
Hi, Tamara. Great talk. And I want to say I think you're exactly right about the budget constraint. That is very often how at least we design the experiments, and that's the main kind of dimension. And I wanted to ask you about maybe if you've thought about two other dimensions. The first is, oftentimes we have the choice of doing bulk sequence, and I'm thinking of the cancer. You kind of switched from population genetics to medical genetics. So I want to focus on the TCGA cancer example, at least temporarily. So oftentimes we have the choice of bulk sequencing or single cell sequencing, or somewhere in between where we do some kind of cell sorting and purification. And so I was wondering if you've thought about ways of incorporating that trade-off, that decision. How many single cells do I want where I get a lot of fidelity about the genome versus bulk sequencing where I get a big sample, but there might be heterogeneity and mixture populations in there. So that's one trade-off. And the other trade-off that I'm thinking about is in kind of medical genetics or TCGA, oftentimes we're interested in knowing something about the population of variants that might exist in a particular type of like solid tumor, like breast cancer or something like that, that exists in the population. But we also might want to know about what's in this individual. And so we can target the treatment to the individual. So we want to trade off between accuracy on the individual versus accuracy on the population. And I wonder if you've thought about that.
First of all, it is really great to hear that everything I said wasn't totally dumb from somebody who I think is much more an expert in these things. So thanks so much for chiming in. So the short answer to everything that you said is no. I have not thought of anything that you talked about. It all sounds super awesome, and I would love to chat more. And then let me just give you my initial thoughts based on everything that you said at this moment, but like this should definitely be double checked. So in the first case, in terms of making these decisions between like essentially like, as I understand it, different types of sequencing that you could do, it seems to me that, at least based on my understanding of your description, that this could be conceptualized as perhaps like an additional discrete trade-off. Like if you could put each type of sequencing separately into our model, then you could get a prediction for the number of new variants you would get with the different types of sequencing. And then you could compare those. You could say like, oh, in this one, I'm going to get this number, in this one, I'm going to get this number. And so the way that I'm going to choose the type of sequencing that I'm going to do is based on comparing those two numbers directly. I'm sure, I just feel like in everything I touch in biology, there's so much more nuance than I anticipated. And so I feel like to really get that to work, there's probably some thought that would have to go into it. But at least on the face of it, it seems like if you had a discrete number of different methods that you could consider that separately could fit into the model, that you could come up with like an appropriate lambda and appropriate phi and so on like that you could do this comparison. And then two, in terms of trading off wanting to learn about the individual versus wanting to learn about the population, this also seems like something that you could fit into the framework in the following way, although I think it would be challenging. So what I think you have to do is come up with an optimization objective that describes that trade-off. And so here we said we want to learn the maximum number of variants. But in this ongoing work that I mentioned at LMRL, what we do is we just change the optimization objective. We say, oh, the objective is the power in our statistical test. And so something that's really nice about the work is that like you can basically just put in a new objective. And so it's very flexible that way. And to the best of our knowledge, that doesn't really come up elsewhere in biology. There's some work in variant association studies where there's like a particular burden test that they look at and they look at a prior analysis, but it seems like it's all very formulated to that burden test. And I think something that's nice here is that it seems like very agnostic to the objective. So you can put in a new objective. And so you can come up with an objective that says, okay, well, I want some balance between the number of variants or whatever I want in the population versus the number of variants or whatever I want in the individual. And then I think that the challenging part is that we always act like we can just choose these losses, and then like actually choosing that trade-off, I think, is challenging. But if you're willing to commit to like an actual number, like here's how I'm gonna trade them off, like every 2 new variants in the individual is worth 1 new variant in the population, then if you're willing to commit to that, then I think you can just use the whole setup because you just have a particular objective. Again, like all of this is off the top of my head, so like we want to super double check it. But this is so interesting. I mean, I'd love to follow up with you sometime in chat if it's of interest.
Okay, so let me see if we have any other question, comments. We don't. So let's thank again to Tamara for this amazing presentation.
Thanks so much, everybody. Thanks for having me out and all the great questions. Really appreciate it.
Stop.